Normally in organisms like us or fruit flies or other animals, only one of the parental genes gets moved to the next generation. So that in our children, you have one gene from mom, one gene from dad. But when you do active genetics, you can transmit both copies of the parental gene or the maternal gene to the progeny.
With active genetics, we're capable of modifying one of these genes so that it cuts the other one and copies itself over. So now you don't have, even if you inherit one from mom and one from dad, you're actually gonna be giving only one specific copy, the one that is active, to your kids.
So there's a vast array of organisms that are out there for which the genome sequence is known, but for which genetic tools are not yet available. So what Valentino had come up with was a way in a single swoop to make the effect visible immediately in the first generation.
But there were not enough tools to work in these other species, and that's how we came with this idea of these genetic elements that can convert the other chromosome, because they come very useful when you're trying to generate mutations into other species or carrying characteristics that you want generation after generation.
There are huge numbers of possible applications of active genetics. I mean, the most obvious one, of course, and the one that we're focused on the most at the moment, is the one of immunizing mosquitoes and other insects that carry diseases so that they no longer are capable of propagating those parasites. So the malarial parasite, the idea is to make antibodies that recognize it and then make the mosquito have antibodies against it so that it's immunized against the parasite. And to spread those antibodies throughout the population so that all the mosquitoes are immunized and cannot infect human beings with the parasite. So that's like the main goal.
At the level of UCSD, we're really trying to push this technology in other directions so that it would sprout in other fields, and we've been really pushing for the agricultural application, especially because if it did work as efficiently as it does in mosquito and fruit fly, also in plants, that would be a very revolutionary technology.
There's been very few times where I've seen a basic research discovery that you can see have the possibility of paying off rapidly. So this is something that could be taken from working with fruit flies and mosquitoes in the lab to having the possibility of being taken rapidly to the field to improve human health.